Today I'm going to talk about color. Just for those of you that don't know, I'm a big fan of the arts. I draw and paint, and I'm a photographer. And most of that would be pretty boring without color. So I'm going to talk about how the human eye detects color. So basically, light enters your eye, and it hits your retina at the back, which is loaded with two types of photoreceptors. There's rod cells and cone cells. The rod cells are sensitive to different light intensities, and they are sensitive enough to respond to a single photon. And so these are primarily for night vision or lower light vision, as well as helping determine different light levels when you're walking around in the daytime. The cone cells are what detect color. They're less sensitive to light. They're very sensitive to different color wavelengths. And so they have faster response times than your rods, and they help with fast-moving objects and things like that. So typically there's three types. There's short, medium, and long, named after the wavelengths that they correspond to. And how we actually interpret colors is you can see the curves over here on how they correspond to color. And basically what happens is the closer they are to their color, the stronger response they send off to your brain. And your brain interprets the cross-section from two or more of these cone types to determine what color an object is. So, however, some people have problems seeing color, like myself. And typically when people hear color blindness, they think one thing. They think of black and white TV, which is how people with monochromacy would see the world. So this can actually occur several different ways. It's an extremely rare condition. So most people with color blindness actually just have like a color confusion. So they don't see one color or something like that. So chromatopsia is when you only have one cone type or no working cones at all. If you have no working cones at all, your vision is basically just your rod cells. And so there's a whole bunch of light that you can't detect, and it makes your vision extremely poor. And then multiple types of dichromacy at the same time can also cause your vision to appear this way. So dichromacy is when sufferers only have two types of cones. So like I said before, your brain interprets the signals from the cones and kind of uses them in conjunction to tell what color something is. So if you have dichromacy, you typically have a gap somewhere. Protanopia is lacking a long wave cone, so you would have problems seeing red light. Deuteranopia is medium, which is green and yellow lights. And the short wave cone is for blue and violet lights. Anomalous trichromacy is the most common form of color blindness. This is where just one cone is mutated, so it's shifted on the light wavelength on what it picks up. So I'm going to go through the protanomaly, deuteranomaly, and tritonomaly. Protanomaly, this is what I have. It's an anomaly in the L cone. So basically, I see reds differently than all of you. So people with this are less able to discriminate colors. They don't see mixed light as the same colors. Mixed light would be if I have a yellow light and a red light pointed at something, that actually combines to make green usually. So I would see those objects differently than you. and then differently than if there was just a green light pointed at that object. And then there's a darkening at the red end of the spectrum that just makes vision darker, I guess. Deuter anomaly is the most common form. The green cone is shifted toward the red, so darker green objects may appear black, and other hues seem to be shifted towards green, so you pick up a lot more greens when objects are red or orange. Unlike protons, there's no brightness problems. And with triton anomaly, it's caused by a different chromosome. This is chromosome 7, so it's equally rare for males and females at 0.1%. And I'll talk about the other chromosomal causes of the other types of color blindness in a minute. Ishihara testing is pretty much the standard for colorblind tests. I'm sure all of you have seen this at one point. They typically do it at a young age, like 3 to 5. And this is plate number 1. It's just a default to show you what the test is going to look like in case you are colorblind, so you know what to look for. So everyone should see a 12. If you can't, you need to get your eyes checked. This is Ishihara plate number 13, which you should see a 6 on here. Does everybody see a 6?
I thought it was an 8 too.
You might have some trouble. So to me, this is actually just a bunch of dots. I can't make out anything. And then this one, you may or may not be able to see what symbol this is. It's a number 2. And I included this one because it's interesting. People with color blindness pick this up a lot easier than those without. And some people that have normal color vision shouldn't be able to see this at all. But so now I want to talk about the causes of most color blindness. So about 8% of men are colorblind, and only around 0.5% of women are colorblind. And this is because it's related to the X chromosome, which means women typically can carry the gene, but they have 2 X chromosomes, so they can make up for it with their genes. This leads to something interesting because some women can be tetrachromats, which means they inherit a set of mutated cones and a set of normal cones, and they end up with 4 types of cones, which gives them even more overlap for their brain to distinguish color. So it may or may not let them distinguish more colors because that's part of your brain that would need training to distinguish colors. And it's interesting because I come from a family of 4, and my brothers are both colorblind. And so it's possible that my sister is a tetrachromat. And I gave her a color distinction test, and she actually did really well. They mark you 0 to 100, and she scored a 12, 0 being the best. I did about a 50. My other brother scored 120. So I would attribute the score of a 50 just from my general interest in art. So that's the thing, though. With color, it's subjective. It's very subjective because all of us see differently, those with color blindness and without. So that's why I think that art is interesting, because it lets us communicate and then reinterpret very different things. And then we have different names for colors. A lot of different objects could be called red, but there are many different shades of red. So that lets me adjust quite easily because I can pick out colors. I can see stoplights just fine, so don't worry about that. In some countries, you can't drive if you're any type of colorblind, which I can kind of understand, but I can see stoplights fine. So the Himba is a tribe in Angola, and they actually have very different words for color. Well, than we do typically. So Zuzu is dark shades of blue, red, green, and purple. So it's really kind of all over the map as far as the wavelengths of light. Vapa is white and some yellows. Buru is some shades of green and blue. And Dambu is shades of green, red, and brown. So this might mean that they could all be colorblind and are just basing their decisions on what value they see as far as grayscale. But it's just an interesting example of how people see and catalog color differently. Some frames of reference that help adjust with color blindness. Oranges are the best thing ever because they're named for what color they are. So, I mean, I have orange. I got that one down. So it doesn't matter what that would actually appear to me as because everyone calls it orange. So I can adjust the rest of my colors to match. HTML color tables are quite awesome because I can just type in a number and then I know what color I'm getting. Same thing with Pantone matching. It's just a way to match a number to a color, which I've done in art before, painting. If I want a certain color, even though it might be interesting if I pick it out from a color picker, I can pick a number and actually get exactly what color I want. So I ran through that pretty fast, but any questions?